systems reduces effluent disposal. And what are the disadvantages of enzyme immobilization? Uh, Here we have some disadvantages of enzyme immobilization. It gives rise to an additional bearing of cost. Actually, when comparing with the cost, enzyme immobilization, once we will done with the enzyme immobilization, that we can use after several years also. So it gives rise to additional bear on cost, but still we can store it for a longer period. In invariable affects the stability and the activity of the enzymes, and the technique may not be proved of any advantage when one substrate is found to be insoluble, and certain immobilization protocols offer serious problems with respect to a diffusion of substrate to have an access to the enzymes. Here we'll see the enzyme cell immobilization methods. Here you can see there are different stages and there are different types of enzyme immobilization. Here you can see the cross-linking and covalent bonding, entrapment and encapsulation and absorption, cross-linking. First, this we can call like physical methods for immobilization. When coming to the physical method of immobilization, first one is absorption. Here you can see the absorption. This one, absorption. In absorption, it involves the physical binding of enzyme on the surface of carrier mixture or carrier matrix. And carrier may be organic or inorganic. The process of absorption involves the weak interactions like van der Waals bonds or hydrogen bonds. So here the carriers are silica, bentonite, and cellulose. For example, catalase, an enzyme, and invertase. Here, catalase, and which acts as a free radical. Here again, absorption. It is a method that involves the physical binding of enzyme on the surface of the carrier matrix. The carrier may be organic or inorganic. The process of absorption involves the weak interactions like van der Waals or hydrogen bonds. Here, the carriers are silica, bentonite, cellulase, etc. The enzyme is catalase and invertase. And the chemical method of before going to chemical method, here what are the advantages of absorption method? Here, absorption method, when coming to the advantages, absorption method has it is a simple and economical and limited loss of activity and can be recycled, regenerated and reused. When coming to the disadvantages, it is a relatively low surface area for binding, exposure of enzyme to microbial attack, yield are often low due to the inactivation and desorption. So the next one is entrapment. Here you can see the entrapment. Name itself indicating like entrap. Means the enzymes or cells are not directly attached to the support or surface, but that can be simply trapped inside the polymer matrix. Here you can see the structure. Enzymes are held or entrapped within the suitable gels or fibers. It is done in such a way that to retain protein while allowing penetration of substrate. It can be classified into lattice and microcapsule types. Inclusion in gels like polyacrylamide gel and polyvinyl alcohol gels, and inclusion in fibers like cellulose and polyacrylamide amide gels, and inclusion in microcapsules, polyamine and polybasic acid chloride monomers are the entrapment. Then when coming to the lattice type entrapment, here, yes, lattice type entrapment that involves entrapping enzymes within the interstitial spaces of cross-link. Here you can see cross-link, a water insoluble a polymer, some synthetic polymers such as polyacrylamide and polyvinyl alcohol, etc., are natural polymer, that is the starch, have been used to immobilize enzymes using this technique. And microcapsule type entrapment. Here in microcapsule type entrapment, the encapsulation involves enclosing the enzymes within semi-permeable membrane, uh, semi-permeable polymer membranes, that is semi-permeable collidon or nylon membranes in the shape of spheres. What are the advantages and what are the disadvantages of this encapsulation? Here, the main advantage is here we are not using any chemical and no chemical modifications required for this advantage. For this is an best advantage. And disadvantage is the enzyme may leak from the pores and relatively stable forms, and this is very easy to handle and we can reuse this reaction again. Before going to the further, here we have to know the covalent binding. Here, covalent binding. Here I want to show you the structure. This one, covalent binding. Based on the binding of enzymes and the water-soluble carriers by covalent bonds, the functional groups that may take part in this binding are amino group, a carboxyl group, and sulfhydryl group, hydroxyl group, imidazole group, phenolic group, thiol group, and there are many other. The advantage of this covalent bonding enzyme cell immobilization is the binding force between the enzyme and carrier is so strong that no leakage of the enzymes are occur even in the presence of substrate or solution of high ionic state. Then coming to the disadvantage, the covalent binding may alter the conformational structure and active center of the enzyme, that resulting in major loss of activity and changes of the substrate. Then cross-linking. Here you can see the cross-linking. A cross-linking which involves intermolecular cross-linking of enzyme molecules in the presence and absence of solid support. The method produces a three-dimensional cross-linked enzyme aggregate insoluble that is insoluble in water by means of a multifunctional reagent that links covalently to the enzyme molecule. So the advantages of the covalent bonding, uh, the enzyme that strongly bound, that shows like a very little disruption, and this is highly stable reaction, that is a pH, ionic strength, and substrate concentration. Everything will be like highly stable one. When coming to the disadvantages of the cross linkage or cross linking, the cross linking may cause a significant changes in the active site that leads to the changes in the enzyme activity and a rate of the reaction that also affects the yield. And this is not cost effective one. So this is about the types of enzyme immobilization or cell immobilization methods. So yes, we have these many advantages and disadvantages. Like absorption has its own advantages and disadvantages. Cross-linking also just now we discussed, and covalent bonding also has its own advantages and disadvantages. Entrapment and encapsulation also. Along with that, we have some limitations of enzyme immobilization. First, the cost of carriers and immobilization um, is one of the first limitation, and changes in properties. This is depending upon the it is like a selective one, and mass transfer limitations, and problem with the cofactor and regeneration, and problems with the multi-enzyme system, and activity loss during the immobilization. So just now, um, first we have to know the applications. We already discussed like the major applications of the immobilization of enzymes is the production of antibiotics and penicillin um, is the major antibiotic which is have the broad spectrum, which is the first broad spectrum antibiotic, and biomedical applications to overcome the metabolic deficiency with the use of encapsulated enzyme, and pharmacy in the pharmacy applications also. This technique is used in the production of syrups, usually the general example like aristocrat enzyme. It is aristocrat syrup. It is a multivitamin and enzyme-based syrup that enhances the digestion in the kids as well as adults. And production of amino acids. Yes, this enzyme technology is useful in the production of multiple amino acid production. This is about the enzyme technology and enzyme immobilization. Next, our topic is Our next topic is biodegradation of plastic. Here, we have discussed about like um, like pollution factors during the sewage wastewater treatment. So now, not only India, around the world, plastic have become an essential part of our modern society. Many of the products used in our daily lives are made up of plastic. Then the plastic are crucial in various industries like transportation, food, transportation, food, healthcare, and energy. Then a population growth and increasing demand for the goods that have led to a substantial increase in the plastic production, that leading to the generation of massive quantities of plastic waste and environmental concerns, that including the emission of greenhouse gases. Only around 9% of the plastic waste produced, that is recycled, and causing the remain untreated plastic waste to accumulated in our lands and oceans, that is water bodies. Water bodies. This accumulation is causing a severe harm to the environment, plants, animals, and aquatic life, as well as posing risk to human health. In order to address all these challenges, it is necessary to explore and implement an alternative and substantial sources or methods where non-renewable resources use is minimized and materials are reused and recycled. One promising approach that involves using biomass as renewable raw materials. This type of plastic is known as bioplastic. Here you can see the type. Bioplastic. This bioplastic is a polylactic acid. This bioplastic are alternatives to fossil-based conventional plastics that are made up of from renewable sources like plant biomass. They are often 100% bio-based, that are considered sustainable as they reduce a reliance on fossil resources that introduce eco-friendly disposable options and useless toxic production methods. So again, bioplastics. Bioplastics are sustainable alternatives to the traditional plastic that made from renewable resources are that can be biodegradable or are manufactured through the biological process. Simply, bioplastic that refer to plastics derived from the plant or biological source. Here it is the solution for all the problems up to now we discussed, that is bioplastic. According to European bioplastics, and bioplastics that can be defined as having one or both of these traits, it's either bio-based or biodegradable. So being bio-based means that the main source of the product is biomass, primarily a plant such as corn and sugar cane. Biodegradation is a process where microorganisms in the environment that convert the products into natural substances such as water, carbon dioxide, and compost. It's important to understand the bio-based and biodegradable are not the same. The biodegradability is related to the chemical structure of the material, not the source of the product. This means the bioplastics can be entirely bio-based and still not biodegrade, while some fossil-based plastics are biodegradable. And some biodegradable plastics, despite being derived from fossil fuels, are also sometimes labeled as bioplastics, which is discouraged to avoid confusion in the definition of the bioplastic. Finally, bioplastic means a plastic that derived from the plant or any biological source. Here we have types of bioplastics. Before going to the biodegradation, here, yes, we have types of bioplastics. The two major types of bioplastics produced in large quantities are polylactic acid, that is called as PLA, and polyhydroxy alkanoate, that is PHA. Besides these, there are several other bioplastics such as cellulose-based, starch-based, protein-based polymers, and PLA, polylactic acid. The polylactic acid bioplastic is derived from the sugars found in crops like corn, cassava, or sugarcane, its basic binding blocks are lactates or lactic acid monomers, which are polymerized to create PLA. A PLA has numerous advantageous characters that including ease of fabrication and biodegradability and non-toxicity. Where PLA undergoes a biodegradation, it releases carbon dioxide, water, and decomposed organic matter, which can be used by green plants. That polylactic acid plastic is commonly used in green packaging for fresh food products and also has applications in sectors such as construction, agriculture, transportation, electronics, household goods, and textiles. And the other one, second, as I already told you, there are two major types of bioplastics. One is just now we discussed polylactic acid and polyhydroxy alkanoate. This polyhydroxyalkanoate is a thermal plastic, a polyester created from the polymerization of 3-hydroxyalkanoic acid monomers. The PHA is made by microorganisms. These microbes synthesize PHA as a means of storing energy and carbon intracellularly. This PHA has a wide range of applications, particularly in biodegradable implants, drug delivery systems, and tissue engineering, and anti-cancer and antibacterial treatment. A PHA is a biodegradable and harmless to living tissue, such so that it is commonly used in the medical practices like medical application. It also has applications in single-use food package. So here the major uses and disadvantages of a conventional plastic. What we can say like a conventional plastics versus bioplastic. The main is source. Here the conventional plastic that is derived from fossil fuels and petroleum based resources. When coming to the bioplastic that is produced from natural renewable resources and resource availability, this is reliant on finite fossil fuel resources and bioplastic utilize renewable resources and biodegradability. This is a major important factor for us. Conventional plastic that contributes to the environmental pollution. So biodegradability, most conventional plastic are non-biodegradable that leads to environmental pollution. Bioplastic often considered more environmental friendly. So many plastics are biodegradable. And common applications of conventional plastic is used in packaging bags, bottles, construction, textiles, electronics, and there are more. And common applications of bioplastic is applied in biodegradable food packaging, automotive parts, biomedical tools, and there are many. When coming to the examples of the materials of conventional plastic is polyethylene, terephthalate, and polyvinyl chloride, PVC, and polystyrene. These are the examples of conventional plastic. Whereas non-conventional plastic, just now we discussed about PHA and PLA, that is polyhydroxy alkanoate and polylactic acid and polyhydroxy urethanes. Urethanes. These are the uh, major differences between a conventional plastic and bioplastic. The main benefits of the bioplastic is bioplastic contribute to a cleaner environment by lowering the greenhouse gas emission. A manufacturing bioplastic typically requires less energy than a traditional plastic, promoting energy efficiency. Bioplastics reduce the dependence on non-renewable fossil fuels and contribute to carbon neutrality by using the renewable biomass resources. Biodegradable bioplastic provide an eco-friendly end-of-life solution and reducing landfills, waste and environmental pollution. Bioplastics do not contain harmful additives like phthalates or bisphenol A, and bioplastics are versatile and suitable for major and various applications from packaging to the medical devices and offering a wide range of sustainable solutions. Then, what are the challenges and limitations of the bioplastics? While bioplastics are eco-friendly and these are the alternatives, they do not have their limitation and despite their potential benefits, the widespread use of bioplastics faces numerous challenges and a production of bioplastic requires the extensive land resources for cultivating the plants for raw materials, which adds pressure on both the environment and the food supply. Similar to the conventional plastics, bioplastics also raise concerns about chemical leaching and the effectiveness of bioplastics depending upon the factors like chemical composition, manufacturing processes, and end of life management. The use of bioplastics have hidden environmental costs such as the use of toxic pesticides on crop and carbon dioxide emissions from harvesting equipment. Many bioplastic manufacturing facilities rely on fossil fuels to power their operations, consuming nearly as much energy as conventional plastic production. The utilization of edible Biomass raises ethically concerning regarding the potential competition with the food production of more efficient methods for using biowaste must be developed. What are the future aspects of bioplastic? Before going to the future aspects, here we have a slide. Just we already discussed about it emits methane, carbon dioxide, H2O2 and other residues, and CO2, H2O2, other metabolic products under aerobic condition. Here, as we know, microorganisms like bacteria have two types, anaerobic and aerobic. Under the anaerobic conditions, the creation of extracellular enzymes from the microorganisms that entrap and the enzymes that attach to the polymer and split the polymer chain that can able to produce the oligomer, dimer, and the monomer. And the microbial degradation happens under aerobic conditions that releases carbon dioxide, H2O, and other metabolic products. And here we have the fungi, since it's like a creation of plastic bags from a problem regarding its manipulation of enzymes that is ended up in landfills. So to overcome that problem, here we have the bioplastic is produced from the white rot fungi. How to degradation or deterioration works, pH, temperature, and the structure of the plastic lignolytic enzymes that is produced from the fungi, that enzyme excretion from the white rot fungi cell that deteriorate the plastic into lignin proteases. Here you can see in the structure the white rot fungi could be acts as a alternative and in conditions to degradation or deterioration have been made by multiple treatment methods. Here, the pre-treatment happened at a high temperature levels, exposure to the fungi. Here you can see how the enzymatic plastic degradation is happening. The abiotic factors like sunlight, temperature, when it is a biodegradable plastic, the microbial attachment takes place. The enzymatic attack that defragment the plastic into oligomers, dimers, and monomers, that is called as biofragmentation. The biofragmented products like oligomers, dimers, and monomers that can be uptaken by the microorganism, that is assimilated and become like a mineral a mineralization happen. In the last slide, we already discussed like anaerobic and aerobic. So it is released to the waste is released to the environment as like a CO2, CH4, and H. And what are the future uh, what are the factors that affects the biodegradation? That is exposure characteristics like abiotic and biotic and polymer characteristics. Here the abiotic are moisture, temperature, pH, biotic are enzymes, hydrophobic, and polymer characteristics like molecular weight, size, and shape, additives, biosurfactants. Finally, a bioplastic future perspectives. A bioplastic are continually evolving, and the researchers are actively working on the development of the next generation of bioplastics. There are ongoing focuses on developing factors such as recycling method, cost, durability, and biodegradation. There is a one project that is the CIRSI is one innovation bioplastic project that uses engineered microbes to produce biodegradable plastic, unlike traditional bioplastics made from crops. Since these microbes feed on carbon dioxide and hydrogen gases to create eco-friendly polymers suitable for various products from packaging materials to cosmetics. And SHIRK is one innovative innovation that drives its base material from shrimp cells that is made up of by chitosan and the silk protein fibroin. These innovations represent a promising steps toward more suitable and sustainable bioplastics. A future of the bioplastics will resemble the traditional plastics but with a reduced environmentation from edible first generation biomass to non food crops. More efficient methods for using a second generation biowaste must be developed while advan- advancements are still under the development and yes, every researcher and the innovators can contribute by reducing the disposable plastic use that directly impacts on the society. This is about biodegradation of plastic. And our next topic is the bioremediation. Yes. Here, bioremediation. Yes, bioremediation that refers to the process of reduction, elimination, alteration, and transformation of contaminants present in the natural environment like soil, sediments, air, and water through the application of microorganisms like fungi, green plants, and their enzymes. And we can say like it is a waste management technique that uses naturally occurring biological organisms to break down the hazardous substances to a less toxic or non-toxic forms. Bioremediation that includes a series of redox reactions for the production of energy within microbial cells for cell maintenance and reproduction. A biodegradation is a global, regional, and local application for removing the pollutants from the environment or restoring the contaminated site. So, biodegradation to the process of using microorganisms to remove not only the microorganisms, living resources to remove the environmental pollution or prevent the environmental pollution. And yes, there is a type of there is a um, other names for the bioremediation is biotreatment, bioreclamation, and biorestoration. And xenobiotics that broadly refers to the unnatural, foreign, and synthetic chemicals such as pesticides, herbicides, and other organic compounds. Here, bioremediation is are of different types like biostimulation, bioaugmentation, and intrinsic bioremediation. So bioremediation, it is a triple corner process where biopollutants are of two types, organic and inorganic. Organic are solid liquid gases that is released to the environment, reaches to the organism. So here the environments like the soil, water, air, and organisms here are microorganisms, plants, enzymes. The monitoring of soil physically and chemically is a time consuming processes in order to measure the pollution of the soil after contamination in a short period of time. Microbial and biochemical properties of the soil are to be determined. The biodegradation of the bioremediation process depends upon the different factors for the removal of the contamination. And here, the concentration of the contaminants, like the concentration of the contaminants that are uh, directly affects on the microbial activity, lower the concentration of the contaminants, there will be a decreasing rate of degrading enzymes produced by the bacteria in the soil. Toxic effects are observed in the presence of high concentrations of the contaminant. The decomposition rate of catabolic enzymes can be increased by the synergistic reactions between the different components of the contaminants. And in terms of soil contamination, uh, nutrient availability like carbon, nitrogen, and phosphorus, potassium, calcium are the basic requirements for the growth of microorganisms. The concentration of the nutrient availability is directly affects the degradation of the contaminant. Um, so here, anaerobic or aerobic metabolism of the microorganism that involves oxidation and reduction reactions for detoxification. Oxygen could be reduced to water and the oxidized organic compound. Anaerobic reaction can use the nitrate. In turn, biomass is gained from the bacterial or fungal growth. In many cases, combined effects are needed. Indigenous microbes found naturally in polluted sites. So, the characteristics of the polluted sites, like a contaminated soil, is the bioremediation process is significantly affected by the different parameters of the contaminated soil, such as pH, temperature, uh, permeability, and water holding capacity, temperature, and oxygen availability. A pH that required for the bioremediation process, which ranges from 6 to 8, and the neutral pH is suitable for the biodegradation of the petroleum hydrocarbons, whereas some fungi are acidophilic microbes degrade contaminants in an acidic environment. Temperature that affect the biodegradation of contaminants is also affected by the temperature, especially in case of hydrocarbons under both in situ and ex situ conditions. It has been found that the higher temperatures like 30 degrees centigrade to 40 degrees centigrade, increasing the bio remediation in the soil as well as in the marine environment. Next, coming to the oxygen availability. Oxygen, it is a very important factor to determine the extent, rate of biodegradation of contaminants. Aerobic biodegradation is much more faster than the anaerobic biodegradation. For the aerobic respiratory breakdown of organic contaminants, oxygen availability plays a significant role in the majority of the cases. The addition of hydrogen peroxide is used to introduce the oxygen. Hydrogen peroxide is about 7 times more soluble in water than the oxygen. So here we have two types of bioremediation processes. One is intrinsic and another one is extrinsic. That is in situ bioremediation and ex situ bioremediation. Uh, what is in situ bioremediation? In situ bioremediation is the in-site treatment of contaminant using biological agents. It is a cleanup approach between microbes and contaminants directly and for biotransformation. Yes, in situ bioremediation is also of two types. One is and intrinsic and engineered bioremediation. Intrinsic bioremediation or natural attenuation is a passive process of treatment of polluted sites without any human intervention through naturally occurring microbial population. It is a natural remediation of environmental pollutants into non-toxic or less toxic forms using inherent capacities of microorganisms without taking any engineering steps to enhance the process. The major applications of intrinsic bioremediation is for controlling of contaminant at waste sites. This type of remediation requires monitoring of the role of native microorganisms in eliminating contaminants via tests performed at the field sites or on-site derived samples of the soil, sediment, or water taken place, and different environmental factors that favors the intrinsic bioremediation, such as pH, concentration, temperature, and nutrient availability. It is a cost-effective method which has the limitation is that no longer time to achieve the target levels of the pollutant concentration. And the other one is engineered bioremediation. Engineered bioremediation or accelerated bioremediation is the advanced process of applications of engineered systems to stimulate microbial activity for uh, remediation of environmental pollution, where intrinsic bioremediation isn't a feasible uh, accelerated bioremediation is proceeded where either a substrate or nutrients are added to the environment to degrade the toxicity of contaminants that making the microorganisms that grow more rapidly. This process that accelerates the biodegradation process through the optimization of physical and chemical conditions that vitalizing the growth of microorganisms. Usually, the microorganisms in the ingredients, uh, but occasionally microorganisms that are very efficient at degrading a certain contaminant are additionally added. And engineered bioremediation may be chosen over intrinsic bioremediation because of time and liability. The limitations of this engineered bioremediation is the ineffective reactivity with the metal contamination that are mixed with organic compounds. Next, in situ bioremediation. As we already discussed in our first slide, like biostimulation, bioaugmentation, and intrin- uh, intrinsic bioremediation. Yes, all these comes under in situ bioremediation technique. So, first one is bioaugmentation. Here, the bioaugmentation here the bioaugmentation So, bio, side for bio. Uh, Bio augmentation. It is a process of addition of the culture microbial population which have the ability to degrade a specific soil and groundwater contaminant. Um, a technique of bioremediation in which the strains of natural or genetically engineered bacteria with unique metabolic profiles are added to the contaminated site in order to supplement the indigenous microflora and speed up the reaction, speed up the biodegradation process. And some common applications involves in the bioaugmentation for a chlorinated contaminants, a petroleum hydrocarbon, etc. The microorganisms are isolated either from contaminated sites, historically sites, or genetically modified to support the bioremediation process of the contamination sites. Here, the petroleum and its byproducts are hydrocarbons. Oil constitute a variety of hydrocarbons like the xylene, naphthalene, octane, camphor, etc. The pollutants can be degraded by a consortium of microorganisms. Those are Pseudomonas, Corynebacteria, Arthobacteria, and Mycobacterium, and Nocardia. Here, genetically engineered bacterial strains are used to enhance the bioremediation process. In the year of 1979, Anand Mohan Chakrabarti obtained a strain of Pseudomonas putida that contained XYL and NaH plasmid, as well as a hybrid plasmid derived by recombinating parts of CAM and OCT. But in 1990, the USA government allowed him to use the superbug for cleaning up of an oil spill in water of state of Texas. It was produced on a large scale in the laboratory and mixed with straw and dried. And biostimulation, microbes cannot use pollutant as the only source of energy. Thus, they need to be assessed with supplied nutrients. Biostimulation is the process of environmental modifications via addition of limiting nutrients and electron acceptors like phosphorus, nitrogen, oxygen, or carbon uh, uh, in order to stimulate the existing microbial population, which are involved in bioremediation. It is most common remediation approach against petroleum pollutants in the soil. Uh, and another term that is bio-slurping. Slurping means a multi-phased extraction is the process of a in situ aerobic bio remediation of contaminated soils using the bio venting and vacuum enhanced pre-product recovery the extracts like light and um, no aqueous phase liquids like from the capillary fringe, and bioslurping is limited to 25 feet below ground surface, as the contaminants cannot be lifted more than 25 feet. By this method, the effective for removing a free product, that is floating on water tape. And another term, that is biosphering, the injection of gas and gas phase nutrients pressure into the saturated zone, applying pressure to promote aerobic degradation. It is the most recommended approach for aerobic degradation of sites affected with the lighter to heavier petroleum contaminants such as oil, diesel, gasoline, and jet fuels. Lighter ones are removed easily, but heavier ones are to minimum level of microbial bioavailability requires a longer process of treatment. In it, the cost can be reduced by reducing the diameter of the injection point. And bioventing, bioventing, the most common in situ treatment, and involves supply air and nutrients through wells or to contaminated soils to stimulate the indigenous microorganisms. Bioventing is a is applied for bioremediation of petroleum hydrocarbons contaminated in soil through air supply to an unsaturated soil zone using a combination of pumps and blowers for continuous injection of low volumes of the air. It can be categorized like either aerobic and anaerobic or co-metabolic depending upon their amendments used. The slow removal of air and maintaining 5% of oxygen in the subsurface is generally practiced for biovent. Our next topic is phytoremediation. Yes, phytoremediation is the use of plants for accumulation and removal of um, or conversion of pollutants, and it is use of plant its products for the decontamination or stabilization of contaminants and metals from the soil. There are certain varieties of plants which have the ability to vacuum high, high heavy metals through the soil via root and concentrate them in the stems, shoots, and leaves. These plants possess genes that can regulate the amount of metals taken up from the soil by roots and deposited at other local locations within the plants. And depending on the underlying processes, applicability, and types of contaminants, phytoremediation can broadly uh, categorized as like phytodegradation and phytostimulation, that is also called as rhizodegradation, and a phytovolatilization, and phyto extraction, phyto rhizofiltration, and phyto stabilization. Before going to the internal uh, explanation. Here you can see a diagram which shows us about all the types or categories of the phytoremediation in one picture, that like biodegradation and biovolatilization, bioextraction and biostabilization and biostimulation. Approximately 400 plant species have been classified as hyperaccumulators of heavy metals such as grasses, willow, sunflower, corn, hemp, and flax, alfalfa, Indian mustard, and poplar, and water hyacinth. Yes, there are many other plants which can be hyper accumulators of the heavy metals, and like we can see in our daily life. And before um, discussion about this biodegradation, here uh, the root exudates of these plants play an important role in the bioremediation as it activates the surrounding microorganisms. Genetic engineering is a process are used in the case of Bt protein or insect pheromone producing plants to reduce the use of pesticides. And phytoremediation, it is a effective and a low cost environmental friendly soil cleanup of heavy metals and organic compounds. Pollutants are absorbed in the roots, thus plants are removed, could be disposed or burned. Sunflower plants were used to remove uh, cesium and strontium from ponds at Chernobyl, a nuclear power plant. Transgenic plants with exogenous metallothionein, it is a metal binding protein used to remove the metals. And here bioremediation of metal contaminated environment, that is metals bioremediation mechanisms and metal immobilization and the complexation. Metal immobilization is of two types, like complexation, that is bioaccumulation, and bioabsorption, already discussed, and precipitation and solubilization, like a bioleaching, organic acids and siderophores and root exudates. And here we have these many resources uh, for heavy metals in the environment, that is mining activities, industries, agrochemicals, waste disposables, and atmospheric deposition. And other term is biosurfactants. The biosurfactants are the chemical compounds characterized by the hydrophobic and hydrophilic regions in one molecule. Biosurfactants from bacteria, cyanobacteria, fungi, yeast are classified into glycolipids, uh, lipopeptides, phospholipids, glycoproteins, a polymeric biosurfactant. A biosurfactants are producing genetically engineered microorganism is Pseudomonas aeruginosa. This new strain can be produce a glycolipid emulsifier. It can reduce the surface tension of an oil-water interface. The reduced interfacial tension promotes a biodegradation of oils and the bioremediation of industrial waste. Like here you can see the slurry phase bioreactor and land farming bioremediation. That is a variety of pollutants are discharged to the environment from a large number of industries and mills. That is the bioremediation of dyes, bioremediation in the paper and pulp industry. And yes, Just now we have discussed with, different technologies like in situ, ex situ, and bioreactors, and prevention of control of pollution. These activities are supported by a set of legislative and regulatory promotional measurements, such as policy statements on abatement of pollution 1992 and the National Environmental Policy 2006. The major actions of the abatement of pollution and environmental cleanup are follows like a cleaning environment and control of pollution, environmental education, environmental impact assessment, environmental information system, environmental research, forest protection, hazardous substances management, climate change, and clean development mechanisms, that is CDM. And there are immediate necessity for initiating all India coordinating a project on the bioremediation of the contaminated sites involving a premier institutions in this field for conducting large scale demonstration projects like here about the major pilot field scale of a thing is a bioremediation from the lab pilot. Here, day by day, at our labs, our research lab, we use many pilot experiments or many experiments that releases uh, the metals, so that at our lab also we can use like a hairy root system, hairy root production with the cell culture, and um, that can be directly goes to the field. The medicinal plants cultivation, production of variety of valuable metabolites, the hairy root metabolite engineering, optimization of metabolic pathways, utilization into the environment, and increased secondary metabolite production and elicitation, uh, permeability, and entrapping, and environmental cleanup, that is phytoremediation. And other thing is the advantages of bioremediation. Here, the bioremediation is a natural process and is therefore perceived by the public. Bioremediation is useful for the complete destruction of a wide variety of contaminants instead of transferring the contaminants from the one environmental medium to the another. For example, from land to the water or to the air, the complete destruction of target pollutants is possible. And bioremediation can often be carried out on site, often without causing a major disruption of normal activities. A bioremediation can prove less extensive than other technologies than that are used for cleanup as hazardous waste. The disadvantages, yes, it also have disadvantages. A bioremediation is limited to those compounds are biodegradable, not all. And there are some concerns that the products of biodegradation may be